So today we're going to be looking at the structure of nucleic acids. And so nucleic acids essentially hold information for manufacturing functional molecules, proteins, for key processes in a cell. And cells, being the building blocks of life, means that nucleic acids hold the information that encodes for life. So there are two primary types of nucleic acids. There is DNA and RNA. And so what's the difference between these two? DNA is short for deoxyribonucleic acid. And RNA is short for just ribonucleic acid. So, what does this mean? Well, if we come over here, we can see the structure of the sugar that is present in both DNA and RNA. And so, in DNA, there is a deoxyribose sugar, while in RNA, there's just a ribose sugar. What does this mean? So, a ribose sugar is a type of molecule seen here. So both of these are ribose sugars, but the difference is here. We're missing an oxygen in DNA, while RNA has that oxygen. So the deoxy refers to the lack of that oxygen. So the differences between DNA and RNA don't end there. So overall, the structure of a nucleic acid will be composed of a phosphate group connected to this pentose sugar, pent meaning 5, so 5 carbon sugar, with a nitrogenous base, right? And so this is the overall structure. This is what we call a nucleotide. A nucleotide, right? And so many of these come to form the structure that we see of DNA and RNA. So the phosphate of the next nucleotide will attach or form a bond with this carbon right here. And it will go on to continue that pattern, and we'll get the nucleotide sequence that we need. So, and these carbons are numbered from 1 through to 5. This here is an oxygen. So we have carbon 1 through to 5. So 1, 2, 3, 4, and another fifth one right there. So we can, if we come up here, we can see 1, 2, 3, 4, and this fifth one right here. Another difference between DNA and RNA is the fact that these nitrogenous bases in DNA, they are adenine, guanine, cytosine, and thymine. DNA contains these 4 nitrogenous bases. Cytosine and guanine are complementary, and adenine and thymine are complementary. Cytosine and guanine form 3 hydrogen bonds, while adenine and thymine form 2 hydrogen bonds. However, in RNA, the same nucleotides are present, except for this thymine is not present. Instead, we have uracil. And the function of uracil is the same. However, it's just a different nitrogenous base. So that's another difference we have there. So obviously we have DNA. DNA is a double-stranded molecule, and it forms a double helix. So a single helix would be just this sort of shape, a spiral pattern, DNA forms a double helix shape. And it is anti-parallel. Now what does that mean? So it means that this strand, so we have 5 carbons. So we can use those to determine the overall direction that the DNA strand is going. So we have the 5' end, and this will be the 3' end, and because it's anti-parallel, meaning that it's parallel but goes in opposite directions, this will be this strand's 5' end, and this will be this strand's 3' end. So what does this mean? So when we have our pentose sugar, so 5, 4, 3, 3, 3, 2, and 1. This is the 5' end, and this is the 3' end. And it's very important to note that this is the same regardless of whatever nucleic acid you're dealing with. So, DNA, anti-parallel, double-stranded, forms a double helix, and it contains adenine, guanine, cytosine, and thymine. Now, RNA. RNA, there are 3 primary types of RNA. There is mRNA, tRNA, and rRNA. So mRNA, it's a linear molecule, so it's a straight line. It also follows the same 5' to 3' notation that we use for DNA. However, again, so these are the nitrogenous bases there. We have uracil here and not thymine. And so the purpose of messenger RNA is to transport genetic information from the nucleus of the cell out to ribosomes, which are organelles that convert that genetic information into an amino acid chain to form proteins. Now, tRNA. The shape of tRNA is a coiled cloverleaf shape. So that looks something like this. So we have the nucleotide sequences here, all around the inside. And here will be an anticodon, and it's involved in translation of a gene. So translation is the process of converting a nucleotide sequence into an amino acid sequence to create a protein. And it delivers amino acids to ribosomes during protein synthesis after it recognizes specific nucleotides. So this is a recognition site, and it will use the sequence from the mRNA to bring in an amino acid to form a chain. And rRNA, the overall structure of rRNA is a sort of spherical shape. So it's a spherical shape. It mainly is a structural component of ribosomes, as mentioned previously. So ribosomes are composed of protein, and rRNA. And its primary function is involved in the process of protein synthesis. Another thing to note here is that DNA and RNA will always be synthesized in a 5' to 3' direction. And so that's very important because we have our double-stranded molecule here that there's a 5' end and a 3' end, and there is also if we uncoil it and look at it just in a linear fashion, 5' end, 3' end. There is a template strand and a coding strand. And so overall, the coding strand is what contains the sequence of nucleotides that actually codes for the amino acid, but when converting from DNA to mRNA, the template strand is the one that is transcribed because we want the same nucleotide sequences. So if the coding strand was transcribed, we'd result in getting the template strand out, as the process of transcription involves using complementary nitrogenous bases. So complementary nitrogenous bases. So if we had a cytosine, we'd have a guanine instead. And so obviously that's going to result in a opposite nucleotide sequence, and that's not something we want. So the template strand is the one that's going to be transcribed. So overall, two primary types of nucleic acids. We have DNA and RNA. And DNA, double-stranded, has that double helix shape and codes for the synthesis of proteins, or holds the code to create proteins. However, we also have RNA. There are three main types: mRNA, tRNA, and rRNA. And so RNA contains uracil instead of thymine, and is oxygenated, so it has that extra oxygen in its 5-pentose sugar. Genetic code from the nucleus to ribosomes, where it can be transcribed into proteins. tRNA is cloverleaf shaped and takes amino acids to the ribosomes to form a polypeptide chain in the function of creating proteins. rRNA is spherical shaped and forms the structural component of ribosomes. So that's all for the structure of nucleic acids. In this video, we'll next be looking at the process of transcription.